So today, two different things. Well, they're sort of related to each other. So I just want to talk about what is V1 actually doing? So a lot of times, if you take a neuroscience class, they talk about simple and complex cells and blah blah. But what is the orientation part of V1 actually detecting? And so and this is also a way of a simple model of how to make a complex cell. So that's basically what under the topic of Gabor filters, Dennis Gabor. And then I'll talk about sort of aperture problems in general. And so so this falls under the category, I say every year, of solving problems you didn't know you had. So one of the problems is if you make a very fine receptor array that can pick up little tiny features. I mean, like imagine you could make a detector that looks at the whole face. But, in that case, it actually can't pick up small details like the eyes versus the nose. It just sort of picks up a blurry blob for the whole face. If you make small detectors that could pick up, the eyebrow line and the nose and shading, then what's going to happen is you create these things called aperture problems. And what they are basically is the fact that you're just looking at a tiny bit of the world. And so, and these have to actually be solved by by combining information across space. And that's really what the aperture problem is about. It's about how we combine information across space in order to solve these problems that you didn't know you had, because you have these little tiny detectors that are looking like at only 1/3 of the moon. And so that's the aperture problem. And then we'll introduce one of the difficult topics, local and pattern motion. And I'm going to go over it once, and then I'll go over it again in the next lecture to hopefully make it clear. So don't panic with the first go over. Just take it in, and we'll go over it again. Okay. So what's the first topic? So how do we make a simple cell? Or what does a simple cell actually detect, and how do we make a complex cell out of a simple cell? So just to go over again, we said that a simple cell has a receptive field, which is the part of the visual world that causes the cell to become active. That's very small. This is only about less than a degree of visual angle, so a half of the moon or a third of the moon. And then within that, we had these on and off fields that look like this. And so those were, I'll put it in the middle, on and off. And so those were excitatory response to off or excitatory response to on. So excitatory response to brightening or excitatory response to dimming. And how were those made? Those were made by taking these LGN cells, say like an on cell, and projecting that with a bunch of other ones up to make this orientation detector by sort of getting lines of on cells from the dorsolateral geniculate. So this was in V1 interblobs, and this was dorsolateral geniculate nucleus. So that was a simple cell, and so then we said complex cells are sort of more complex. And what do they look like? They look like, let's put them just out here, yeah. So what a complex cell looks like is you can't see any on or off subfields. It just responds, no matter where you test with a little spot, it just responds to both on and off. And but it's still orientation selective. So in other words, it likes a bar this direction, but it doesn't like a bar going in this direction. So So the question is, how can we, what's a simple way of making a complex cell out of a simple cell? And so what I'm going to talk about, like in the case of sort of simple neural networks, is a very simple model. The real thing is way more complicated. I mean, one example is that complex cells get inputs from the geniculate. So that's not in the model. So real life, as usual, is more complicated. But what we want to do is just get some understanding of a simplified version. And as before, you might think, oh, it's not simple, it's harder. No, but the real thing is much harder. So one model of what simple cells are doing is called a Gabor filter. And a Gabor filter is basically something that can be applied to an image to detect local orientation. And what is it? It's basically taking a sinusoid. So this is a sinusoid, and multiplying it by a Gaussian. So what does that mean? So let's take a sinusoid. Let's take for instance, a cosine. So what does a cosine look like? If you plot cosine x versus x. So say this is x in this direction, and this is, cosine x in this direction. If you just plug that into MATLAB or some program and just plot, cosine of x, what does it look like? It basically, it starts at 1 at 0, and then, it goes down and up and down, like something like that. So that's what a cosine looks like. So what is a Gabor filter? A Gabor filter is you take a cosine and you multiply it by a Gaussian. And so now we have to sort of define what a Gaussian is. So here's Gaussian. So, a Gaussian, that's just the normal curve. And, what's the equation for a Gaussian? What's the basic equation? So if you look in a statistics textbook, you got a bunch of normalizing. But, basically what is the equation for a Gaussian? It's just e to the minus x squared. So if you just plot that out, e to the minus x squared, you'll get a Gaussian. So if we multiply those two things together, equals a Gabor filter. So let's do that multiplication. So here we're crossing 0 right there, and here we're sort of like at a negative maximum. Here's a negative maximum, and then here we're crossing 0. So basically, let's start at 0. So at 0, we'll have a big number. And then, since we're multiplying by this sinusoid, it goes down to 0 at this point. So we'll go down to 0 like that. But then we're also multiplying, we're multiplying the Gaussian, and the Gaussian's getting smaller. And so when we reach this negative maximum, it's not as big. And then it looks like that, something like that. So it looks like something that is a basically, is that the 0? Yeah, okay. So it's slightly squished, but so basically it just looks like something that looks like a cosine, but it's kind of a squashed cosine. Like you took a cosine and then squashed it down. Now that describes going across the receptive field that way. Now what about, going across the receptive field this way? That's just a Gaussian. So if we look at the long axis, that's just going to be nothing, and then bigger, 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 and then you get to the other end of it and it goes down. But if we go in this direction, it's sort of going up and down. It's got sort of a peak in the middle, and then two troughs on either side. So that's our model. Now, is that a good model of a simple cell? No, not a very good model. What's the negative business? Cells can't indicate negative. They just fire or they don't fire. And the off field fires excitatory spikes in response to dimming. So it's kind of, we're taking liberties, but it's an easy thing to compute. We just take a cosine and multiply it by a Gaussian, and then we get one of these guys. So that's our model of a simple cell. So this would be a model of a simple cell that looks like this one. So this, we'll label that simple cell number 1. Now, we could also have a cosine, a sine instead of a cosine. So what is, because here's a sinusoid. This happens to be just one of the sinusoids. And what kind of a cell would that be modeling? Well, that would be modeling, a simple cell that maybe had 2 fields. So let's imagine we had a simple cell that had, which way did I do it? Say off and on. So we have an off field here and an on field here. Okay, so imagine we just had a 2-field simple cell. That would be kind of like a sine instead of a cosine simple cell. And so what does that look like? Well, in that case, we would start off with a sine. And what does a sine look like? Sine starts at 0. Same spacing between the humps, but they're offset by a quarter of a cycle. So this is what a sine looks like if you just plot out a sine. And what would it look like over here? It's just off by a quarter of a cycle. So it would be going like this. And then its peak would be right there, something like that. So it's the same as a sine. It's just shifted over by a quarter of a cycle. And so then we would multiply that by the Gaussian. So here's our same Gaussian here. Just it goes up right there, and that equals another Gabor filter. So here's another one. So this one, since we're multiplying by a sine, so this guy is sine of x going in that direction, x going in this direction. So now that means we're going to be 0 because we multiplied by that 0 at the peak of the Gaussian. And then here's where it's going to be the biggest, right there. And here's where it's going to be the biggest negative. So it's going to be the biggest here, like that, and then we'll sort of go like this. And on the other side, it's kind of like a mirror image of that. So there's the biggest, and then it gets smaller like that. So it's the same kind of thing. It's got some ripples in it, but they die out quickly. So this is simple cell number 2. It looks like this one. So here's simple number 1. Here's simple number 2. And so that's simple number 2. Okay, so if we have those two together, then what we can do, especially if we're making a very simple model, is we just square them and add them. So we square and add, and then we get a perfect complex cell. So why are we squaring? Does the brain square? No, the brain doesn't exactly square. It's just kind of handy to square. So, way back in the day, if you remember doing trigonometric identities, there's a trigonometric identity cosine squared of something plus sine squared of something equals 1. So what does that mean sort of graph-wise? Well, it means like, you take a cosine, then you square it. What does that mean? Well, it sort of turns it makes the humps a little bit sharper and turns them all up like that. And then you take a sine and you square that one. Those are offset humps. So it turns all these humps, when you square them, it makes them all positive, a little sharper. And then when we add them together, it equals exactly 1. It gets rid of all the bumps. And so if we square the output of these two operations, then we'll end up with a completely flat cell that responds to little ripples, but it doesn't care where they are. And so this is a very simple model. Like, when you first see it, you say, I don't really understand the Gaussian. It's got multiplying things. What's going on here? But you can sort of just get a feel for it. So if you have this detector or this filter, and what it does is, so this is the filter. Now we haven't talked about like what it's seeing. So once we have this filter, and then we have to actually sort of use it to filter something. So let's put a big green line here. So like, so here's an image down here. And what is the image? Well, the image is, there's a bunch of, brightnesses in the image like that. And so then what we do is we take another copy of that image. Here's another copy of that image. Same thing, supposed to be the same thing. Let's go down below. And then we filter it through this mess. And this is kind of what the cameras in the Tesla do when they're looking out at the world. This is heavily used in machine vision. It took a long time. I mean, the human weasel figured this out in the 60s, but it took a long time before it percolated into machine vision. But it finally did, and then people started this is like a standard thing you do to an image. You make some Gabor filters, and then you filter the image through them. And that's called a convolution. We haven't talked about that. We'll talk about it a little bit later. But it's a way of sort of processing little pieces of the image with these filters. And so what, if you look at this, what is V1 actually detecting? It's not actually detecting edges. It's not really detecting edges. What it's looking for, it will respond to an edge, but what it's really looking for is little ripples in a certain orientation. That's really what it's looking for. Certain orientation and certain spacing. So what is the absolute best thing that a simple cell will respond to? Well, it would, that simple cell would respond to something that's really bright and then something that's dark, and then, it doesn't care outside of that. So that's the kind of, so it would respond to something that's bright, especially bright in the middle, and then it gets sort of darker and darker, and especially dark in the middle of the off fields, and then back up again. So if you went across it this way, what is it really like? It really like something that is, something that is, bright here and dark here like that. So that's really what V1 does. It looks out at the world and it says, like, to what extent at each piece of the world do we have these little ripples, like curtains? You could sort of think of them as, like drapes, sort of undulating drapes. That's actually what it's detecting, what a simple cell detects. It just says, well, there's, and if you give it the wrong spacing, it won't like it, right? So if you give it something that's spaced too far like that, it won't like it as much. And so it's sensitive to the spacing of the ripples too. It's the orientation of the ripples and the spacing of the ripples. That's actually what V1 is doing when it looks out in the world. Now, these are tiny little ripples. So what are we talking about sort of, in real life? Say we have like a smiling face here. How big are these receptive fields? Well, they're just looking at little things like that, like the top of the top of the line of the white of your eye, or the little corner of the edge of your nose. So these are tiny little things. They're looking for that. That's the level of the ripples that we're talking about. These little ripples at the level of sort of a little tiny piece of the nose or a tiny piece of the eye. So that's what V1 actually detects. And when we make a perfect complex cell out of these two simple cells, so when you make this, this perfect complex cell, what do we get? We get something that is now sensitive to any offset of the ripples. So it doesn't matter exactly where the ripples are. If you offset the ripples a little bit, then it's okay. If you make the ripples centered, it's okay. Offset them the other way, it's okay. That's what a complex cell does. So what it's doing is it's saying, I've detected some ripples here, but I don't care exactly where they're placed. It's kind of abstracting away from the ripple detection, the little drape detection mechanism. So that's actually what V1 does to the world. It sort of looks out at the world, filters it, and finds, like, are there sort of any 45-degree ripples spaced about this far apart in this part of the world? Are there any, like, 135-degree ripples spaced at about this far apart in this part of the world? And then it gets that huge data set, and then combines different simple cells to make complex cells. And what the complex cells do is they essentially lose track of exactly where the ripples are. But that's kind of a good thing, because it's sort of generalizing and saying, like, I know there's some rippliness here, but I don't care exactly where it is. I won't get confused if you slightly offset the rippliness. So it's kind of bizarre when you think of it this way, but this is actually what your V1 is doing. The interblob part of your V1 is doing. So it's filtering the world, finding these little sort of rippliness of a certain spacing, and then generalizing across exactly where that ripple pattern is. That's a complex cell. So this is, it generalizes across exact location of ripples. Because the simple cell, if you offset the ripple pattern, suddenly you'll get a different response. And so the complex cell is saying, like, I'm going to be a little more general about it. I'm detecting there's a ripple pattern here, but I don't care. It's not critical exactly where it is. Makes it a little more robust detector. So, like I said, this is like heavily used in machine vision. This is what you do. This is the first thing you do to an image. You filter it through these little sort of ripple detectors that detect orientation of different parts of the image. Okay. So to some extent, that was an aperture problem because the simple cells are looking at these tiny little on and off regions, and the problem they have is that, if you offset the edge a little bit, suddenly the response goes to hell, because it's like the wrong brightness. With a complex cell, we've gotten over that. Now we can say, anywhere in there is okay. I get a good response anywhere in there. I can tell there's some sort of rippliness in this 45-degree angle. And it doesn't matter exactly where you put it, I'll still respond to it. And so that's an example of an aperture problem. We've combined information across space, and we've kind of lost some information about exactly where this little ripple pattern is. But that's a good thing, because that allows us to sort of more robustly detect shapes and not get confused by the exact positioning of an edge in the shape. So it's pretty abstract. You wouldn't normally get this if you took a neurophysiology course, but I think it's important to really just dig down and see what V1 is actually doing. This is what it's actually doing, the interblob part of it. Okay, so that was quite a mouthful. So let's just back up and talk about a simpler example of an aperture problem that doesn't have any Gaussians in it. And that's the problem of detecting what color something is. Now, when you first think of this problem, you figure, well, you've got red and green cones in the retina. It should be relatively easy to just look out in the world and just pick up what color something is. Because, you have red, blue, and green, and those detectors will tell you whether there's red, blue, and green. But it turns out the problem is of detecting what color something is unfortunately dependent on what color you're illuminating the thing with. So if you have something, if you got something like the sun, so here's the sun. Now if we look at the light coming out of the sun, the light coming out of the sun contains all different colors. So if we plot color on this axis, we often plot the wavelength of color. It's like 400 nanometers up to, say, 700 nanometers. That's the spacing between the individual waves in the color, nanometers. So if we plot the spectrum of the sun, it's got all colors. And what are the colors? Well, it's going to, the lower frequency colors would be, red, and then then you get yellow in there, and then green, and then you eventually get to blue and cyan. So blue is a higher frequency color. So that's the sun. So what do we plot here? We plotted this is the spectrum of colors that the sun is giving off. And so the sun is nice because it sort of has all different colors in it. But say we're looking at an object. So how does color actually work when we talk about an object? Well, if we've got, and here's an apple here. It's supposed to be, there's the stem. There's a little leaf coming off of the apple. Okay, so an apple is red. What does that mean? Well, what that means is that the apple reflects red and absorbs other colors. So if we plot the spectrum, if this is a spectrum of of colors, illuminating scene. So what this is the reflectance spectrum. We could also plot the absorptions, the opposite of it, but here's the reflectance spectrum. And so what does it mean for something to be a red apple? Well, it means that it reflects a lot of red, and then it absorbs a lot of green and blue. So this is, this is red here. So it reflects the red. So when we take this spectrum here and we project it onto the apple, and then it bounces off the apple, and then we look at it, so here's our eye, we can say, oh, that's a red apple because, the red got reflected off and everything else got absorbed. Now, that's all well and good, but what if the color, the color of this illuminating color doesn't have all the colors in it? Now you might think, is that really a problem? And the answer is, yeah, it's a really big problem. It's a huge problem. And the example I give every year is, like, I was in London for about 10 years, going to the pub. It's kind of little smoky, orangey kind of lights in the pub, low light, sort of friendly atmosphere. Drink some very dilute, warm beer. So what color are things in the pub? And the answer is they're all orange. So if you go into the pub with a green sweater, it's orange. So why is it orange? Well, because the light in the pub is kind of like in this range of, the light in the pub is like, there's a lot of orange in it, and there's not very much green and blue. So this guy is, this one is the sun, and, this one is a pub. Now, at first, when I tell you this, it doesn't seem like it's actually believable. But if you actually look at a green sweater in orange light, mostly orange light, almost all the color coming off of that green sweater is orange. And that's actually what the retina is detecting. It'll say, like, there's some orange there. Now, what about if you had an orange sweater? Well, you'd get a brighter orange. So definitely an orange sweater would have more orange in it than a green sweater. But if you looked at what's the maximum amount of color, what's the peak of the spectrum, it would be orange. And why is that? Well, you just took this spectrum and you kind of multiplied it by this guy. And so if you happen to have some green over here, well, that green is going to be really down valued because there's not much green in the pub light, but there's a lot of orange in the pub light. And so even though this green sweater might be absorbing orange, nevertheless, it's getting a ton of orange projected onto it, and so it looks orange. Now, back in the day, when we used to walk to school through snow and we had, cameras that used film, this is a very big problem. So if you go into a pub with film that's designed for taking pictures outdoors and take a picture, what does it look like? It's all orangey. And, back in the day, people would put filters on to sort of fix this problem. Now your cell phone just fixes the problem. It just like calculates, oh, there's an orange cast. And that brings up the point, how does your system solve this problem? Well, it has to look across space. So it's an aperture problem. It has to look at a bunch of places in the scene, and then once it figures that out, it says, oh, there's too much orange in this scene. It's all orange. And so we're going to subtract orange from everybody. And so that's done it's not done in V1. V1 sees everything orange. So there's an example where it's like V1 is dumber than you thought. It was looking at these tiny little pieces, and then it's completely confused by what the color of the illuminant light is. So later on, somebody has to put information together across space, And then once it puts it across space, it can figure out there's an orange cast, and it can subtract off that orange cast. And that's what the computer program in your iPhone does when you go into a pub. It sort of is calculating what the overall cast is and subtracting it off so that all the colors come out correct. So that's an example of an aperture problem. Can only be solved. So what causes it? Looking at tiny little bits of the world. If you looked at the whole pub, you could easily tell it's all orange. But if you just look at a tiny bit, you just say this part's really orange and this part's a little less orange. So it turns out this requires multiple colors. So if you've ever seen a sodium vapor lamp, back in the day before LEDs, they used to use these lamps for street lights that have an orange color to them. But if you look at their spectrum, they just have a little spike in orange, and just orange, nothing else. And if you look at your skin or a green sweater under this sodium vapor lamp, it looks really strange. You can't pick out the colors because you don't have enough colors to actually work it out. You need sort of a range of colors. They can be mostly orange, but they have to be at least a couple different oranges. So it turns out all you need is a couple different oranges spikes, and then that's enough information for your system to figure out what the actual color is. But yes, if you've ever been under a sodium vapor lamp, and then you try to sort of like, what color is this? It's like, whoa, everything, like skin looks really bizarre. All the different, it almost looks like things are in black and white when you look at that. And that makes the system fail because it's got to sum across space, and it needs several different colors in order to figure this problem out. So once again, it's kind of like solving a problem you didn't know you had. It's your system is doing it all the time. And what is it trying to do? Well, it's trying to really, before I erase it, it's trying to get this. It wants to know what the object looks like in white light. That's really what the goal of your color system is. You don't care what the object looks like in orange light. You want to know, like, what is the reflectance spectrum of this thing? What does it look like if you gave it white light, unbiased light? And so we can sort of like figure out, what kind of a thing is it? Okay, so that was the aperture problem for seeing green sweaters in pubs, British pubs. So now, let's talk about the dreaded aperture problem for pattern motion. So, how do we detect motion? Well, we suggested something like a Reichart detector, and all that is two detectors that are separated in space, and one of them has a delay. So this guy is a delay. So this is space, and this guy is a delay. And then we just add those together. And we get a detector that can detect motion. And so how does it detect motion? Well, if you put something and move it in this direction, it first stimulates this detector, then it gets delayed. Then after a little bit of a delay, it stimulates this detector. The delayed signal gets added to the non-delayed signal, and you get a big signal. And then if you go the other way, it doesn't work. So it doesn't work if you go that way. So that was our Reichart detector, named after the guy who originally studied flies. Now that sort of seems like it should this is what happens, and we're assuming that this is what happens in layer 4B. That's where these things are made in a primate. So that seems like it should do a good job of detecting motion. Here's our Reichart detector. But it turns out this Reichart detector has got a lot of problems, problems you didn't know you had. And so let's imagine we've got a big object here. So here's a big object. So it's a big filled-in object. and it's moving in some direction. We don't know what direction it's moving in. We just have some Reichart detectors. And so let's put a Reichart detector right here. That's about how big it is. So let's, so this is our Reichart detector right there. So now if you move this thing, say, we don't know whether it's going up or down, but just say generally move it over to the right. You know, what's going to happen in that Reichart detector? Well, let's look at it over time. So here's time, you know, going in this way. So I'll just draw that little Reichart detector hole. And so what's going to happen? Well, you know, it's going to start off with maybe like something like that, and then it'll get a little bit further across, and then it'll get a little further across, and then the whole thing gets filled up. And so this Reichart detector can detect, I think something went that way. So what does that arrow mean? So we have to sort of just stop for a second. And so, you know, like an arrow is a two-dimensional, it's a vector, it's a two-dimensional thing. So that arrow, you can either think of it as, you know, it's got an X and a Y component, or we could think it has an angle, and it has a speed. And that's the way we're going to think of that in polar coordinates. So this guy is the speed. So how big the vector is tells you how fast the thing is going. And then this angle here is the direction. Okay, so we could also think of it in terms of sort of like the X and Y components, but it's kind of more natural to think of it in sort of like, so if you make a longer arrow, that's faster. You can make the arrow go in any direction. So we've got two dimensions to keep track of, the speed and the direction. Okay, so now we're going to make a distinction between thin arrows and thick arrows. Thin arrow and thick arrow. So our thin arrow is going to be what we'll call the local direction. And the local direction is what the Reichart detector tells us. But that might not be the actual direction. And the thick arrow will be the actual direction, the pattern direction. What direction is it actually moving? Now, again, when you first hear this, it's just like, well, it should be straightforward. The Reichart detector said it was moving that way. Isn't that the answer? Isn't that the right answer? Isn't the Reichart detector just telling us that it's actually going up that way? And the answer, unfortunately, is no. So if we draw sort of a line parallel to the contour, so it's possible that it actually did move in that direction. So let's draw a thick arrow there. You know, it's definitely possible it might have moved in that direction. But unfortunately, the Reichart detector gives you a signal that's radically ambiguous. It's ambiguous over 180 degrees. So in fact, it could have been moving in this direction. Now, remember, there's two parts of that arrow. It's got like, it's moving slightly down, that's the direction part, and then it's longer. It's going faster. That's the speed part. But it turns out it could have been moving in this direction, and it could have been moving in that direction. It's like the Reichart detector has no idea, you know, what direction is actually moving. Who knows? It's radically ambiguous. You just don't realize how ambiguous. Now, why does that happen? So when you first look at this, it's just like, it's just, you know, the Reichart detector is just detecting something moving in this direction. How is it so far off? How could it not distinguish these two different cases, like going down versus almost going up? And the answer is, if you just go back to this little Reichardt detector picture, the Reichardt detector just sees a little darkening going through it. It can't tell, it can't pick up any slippage along the other direction, perpendicular to the Reichardt detector. So if we take our let's take our Reichardt detector here, just put it right here. So what could we do? So we could do something like, let's move an edge through it slowly that way. And that's just the little aperture that the Reichardt detector is looking at. But what if we took an edge and just went faster this way? It looks exactly like slower this way. Or what if we went like, faster this way? That looks exactly like slower this way. Can't tell. It radically can't tell. It's got a huge problem. It really can't tell what the direction is over almost 180 degrees. Yeah. Why can't it detect it? Well, just think of here's, just look at the little aperture. So the little aperture sees like a white thing coming across it like that. Okay, now look at the aperture again. It sees a white thing coming across it. It's actually moving this way now instead of so it can't tell whether it's going faster this way or slower this way or faster this way. They all look exactly the same. The speed is the same. The speed of how fast it goes across the little aperture is the same. Can't tell. So basically, it's just a fault of the detector. The detector just sort of looks at a tiny little part of space, and it just sees something going across that tiny little part of space, but it can't pick up any of the sliding motion, and so that means that it doesn't know what direction it's actually going. So if you went into the nervous system and just tested, it would say like, wow, it seems to respond to right really well, but you'd have to test with an edge at all different angles to find out, oh, this thing's really confused. It doesn't really know what's going on. So how do we solve this problem? Well, we just need to combine information across space. So here's another Reichardt detector here. And what did that Reichardt detector pick up? Well, say it picked up a local motion like this. So now, what are the set of pattern motions? So this is, set of pattern motions consistent with one local motion. That's what this whole collection of arrows is. It's the set of pattern motions that could have caused that one local motion. All of these guys. So now we've got another one of those sets down here. And so if we draw a line that's parallel to the contour, it's possible that the object was actually moving in that direction, but it might have been moving in this direction, might have been moving in this direction, it might have been moving in this direction. Sorry, you got that aperture's in the way. So we've got a whole another set of possible directions that it might have been, and you can begin to see what's the way to solve this problem. Well, you just try to find a pattern direction that's consistent for the two. So this one up here isn't consistent. We can't find it over here, but if we look at this guy, this guy right here is about the same. So that's a common one. So by looking at that, we can say it probably was actually going slightly upward, just by combining information from these two Reichart detectors. They have to be looking at different parts of the object with a different contour, because if the contour is all the same, you're sunk. It's kind of like just having one color in your sodium vapor lamp. You can't solve the problem. You need multiple different orientations of the contours in order to solve the problem. So if you just have if your world consists entirely of one giant contour, so say this is your world. It's just one giant contour, and that contour is, moving across. Then you don't know what direction it's moving at all. So you have to have different orientations of the contour in order to solve this problem. So that's basically how you solve it. You just find what are the motions that are in common across all these detectors. And then that'll solve this radically ambiguous problem where we can't even tell within 180 degrees what direction something is moving. So what's a way of sort of graphically drawing that out? We could draw out, the family of local motions. So the family of local motions consistent with one pattern direction, or local directions. One pattern direction. So that's kind of the opposite. That's the opposite picture. Here we said we detected one local motion perpendicular to the contours. There's perpendicular to the contour. We detected one local motion perpendicular to that contour. Now, what are all the different pattern motions that could have caused that? And then there's the whole bunch of them. Now we're doing the reverse, the converse, which is we have one pattern direction. What are all the different local motions that could have caused that one pattern direction? And the way that we can draw this out on a circle. So if this is a pattern direction now we can say, let's get a bunch of thin arrows that could have caused that pattern direction. And of course, one thin arrow might be the actual same direction and speed. That would be like if this object, instead of moving slightly down, was actually moving perpendicular to this contour. In that case, the pattern direction and the local motion are the same. That can be. But this local motion could also indicate that pattern motion, and this one could indicate it. So there's a whole, if we draw arrows out to the circle, there's a whole bunch of different local motions that are consistent with this one pattern direction. And here's another family. So if something was really moving to the right, what local motions would we expect? Well, we might have that same local motion, or we might have a little local motion like that, or we might have this guy or this guy, all of these like that. And now if we look at these two families, we can see, this guy's the same, right? That guy's the same as this guy. And so how do we solve the problem? So we solve the problem by, one, look across space. And then two, find family of this kind of family of local directions consistent with one pattern direction. So find that kind of family. Find family with most votes. Most votes. And what does that mean? That just means, like, obviously, if we get this local motion, just a single local motion, so say this local motion came in. Well, it's consistent with both of these, right? So we get one vote to both of these directions. Now, what if another locomotion came in, like this locomotion? Well, that locomotion is not found down here. And so then this guy would get 2 votes, this guy only has 1. So that's how you solve the problem. You have to look across space, critical, look across space, and then you just find out which of these families of motions got the most votes. And it won't work if you just have one. If there's only one vote, from one local motion, like one aperture, then you're hosed because you can't tell. So you need a bunch of, you look across space, across orientation. So you have to look across orientation. And if you look across space and orientation and then sum up the votes, you'll get the right answer. Now, once again, it requires an object that has different orientations in it. If you just have only one orientation, then you're hosed. So that's the introduction to the problem you didn't know you had. So, consider this thing, like I moved the paper across the board like that. You can obviously see it's going that way. But, consider what the detectors in your V1 just told the rest of your brain. Well, there was a little detector here. And what did that one think? Well, that one thought it was going that way. And then there's another detector over here. What did that one think? So it's going to the right. That one says, no, it's actually going that way. And then, don't forget the ones behind. So here's one behind. So what does that one think as it goes that way? That one thinks it's going that way. And what about one that's down here? I go across like that. That one thinks, yeah, it's going that way. So every single detector in your V1 has the wrong answer. Every single detector, half of them think it's going, up, half of them think it's going down. Completely wrong answer. The actual answer is, it's going that direction. So there's an example. You just I move this paper back and forth. Every V1 cell in your V1 has the wrong answer, but you can easily see that it's going to the right. So you're very good at solving this problem because you want to know what direction stuff is actually going. If it's coming for you, aiming for your face, trying to get out of the way, or you're trying to grab something. So all animals have to solve this kind of problem. But like I said, it's solving problems you didn't know you had. Okay, so any questions about the first run through the aperture problem for pattern motion? So 2 different ways of thinking. We can sort of think of, we made a local detection, and what are all the pattern directions that could have caused that? That's this case. And then this case is, here's a pattern, a real true pattern direction that the actual object is moving. Given all different orientations, what are the possible local motions that are consistent with that pattern direction? Okie doke. So it's surprising when you think of something. So V1 doesn't solve this problem. V2 doesn't solve the problem very well. A lot of cells in MT don't solve this problem. And MT is already the quote-unquote extrastriate body area. So even in an area that's supposed to be specialized for detecting bodies, it still hasn't solved this basic problem. So there's a lot of stages. There's a lot of problems that we've created by looking at the world through tiny straws. That's really the bottom line. It seems like a good thing to do because then you have a nice high-resolution picture of the world, but it creates all these problems that have to be solved by integrating information across space. Okay, stunned silence. That's the first time through the aperture problem, and we'll go through it again, hopefully, to drive home the points, and then we'll talk about higher order aperture problems for more complicated movements like rotations and expansions and all different things like that. Okay, any questions? No? Okie doke.